So for this video, I will be talking about the physiology of circulation. So there are three major types of blood vessels found in the body: the arteries, capillaries, and the veins. The arteries carry blood away from the heart and branches away as they form smaller and smaller divisions. The veins, on the other hand, by contrast, carry blood towards the heart, and so they are said to join, merge, and converge into successively larger vessels approaching the heart. The walls of the blood vessels, except the very smallest, have three distinct layers or tunics that surround the central blood containing space called the lumen. The capillaries, on the other hand, is the site of gas exchange in the tissues. The outermost layer of blood vessel wall is called the tunica externa or tunica adventitia. It is composed largely of loosely woven collagen fibers that protects, reinforce the vessel, and anchor it to surrounding structures. In larger vessels, the tunica externa contains a system of tiny blood vessels called the vasovassorum, which literally means the vessels of the vessels. This vasovassorum nourishes the more external tissues of blood vessel wall, and the innermost or the luminal portion of the vessel obtains nutrients directly from the blood of the lumen. So in terms of relative size and function, arteries can be divided into three distinct groups: the elastic arteries, the muscular arteries, and the arterioles. Elastic arteries are thick-walled arteries near the heart. These are the aorta and its major branches. These arteries are the largest in diameter and the most elastic. Because of their large lumens, they make their low resistant pathways that conduct blood from heart to medium sized arteries. So the elastic arteries are sometimes called the conducting arteries. Distally, the elastic arteries give way to the muscular arteries, which delivers blood to specific organs and tissues. That's why they are sometimes called as distributing arteries. Proportionally, the muscular arteries have the thickest tunica media of all the blood vessels. Their tunica media contains relatively more smooth muscle and less elastic tissues than do elastic arteries have. For this reason, they are the one that is active during vasoconstriction and less distensible, meaning they have the capability of stretching. In muscular arteries, however, there is an elastic membrane on each face of the tunica media. The last type of artery is the arteriole, which is the smallest of all the arteries. The arterioles have a lumen diameter ranging from 0.3 millimeters down to 10 micrometers. The microscopic capillaries are the smallest of all the blood vessels. So their exceedingly thin walls consist of just thin tunica intima or squamous epithelium. At strategic locations along the outer surface of some capillaries are spider-shaped cells called pericytes. These smooth muscle-like cells stabilize the capillary wall and help control capillary permeability. The veins and venous, on the other hand, is called the capacitance vessel because they have the ability to hold much blood in their vessels. There are three major types of capillaries, namely the continuous capillaries, fenestrated capillaries, and the sinusoid. First is the continuous capillary. Continuous capillaries are very abundant in the skin and muscles, and they are the most common capillary found in the body. They are continuous in the sense that their endothelial cells are joined together by tight junctions, providing an uninterrupted lining. However, these junctions are usually incomplete and leave gaps of unjoined membrane called intercellular clefts, which are just large enough to allow limited passage of fluids and small solutes. So, the continuous capillary is the least permeable and, again, the most common capillary found in the body. Next is the fenestrated capillary. These are very similar to continuous capillary except that their endothelial cell is fenestrated. So they are riddled with oval pores and fenestrations. So their delicate membranes or diaphragm usually covers the fenestration. These are seen in the kidneys and small intestine. The last type of capillary is called the sinusoid capillaries or simply the sinusoids. They are highly modified leaky capillaries found only in the liver, bone marrow, spleen, and adrenal medulla. Sinusoids have large, irregularly shaped lumens and they are usually fenestrated. Their endothelial lining has fewer tight junctions and large intercellular clefts than ordinary capillaries. So, these structural adaptations allow large molecules and even blood cells to pass between the blood and surrounding tissue. To sustain life, blood must be kept circulating. By now, you are aware that the heart is a pump, the arteries are pressure reservoir and conduits, the capillaries are site of gas exchange, and the veins are conduits and blood reservoir. Now for the dynamics of the system. First, we need to define three physiologically important terms, the blood flow, blood pressure, and resistance, and examine how these factors relate to the physiology of blood circulation. Blood flow is the volume of blood flowing through a vessel, an organ, or the entire circulation in a given period. It is usually expressed in millimeters per minute, and if we consider the entire vascular system, the blood flow is equivalent to cardiac output, and under resting conditions, it is relatively constant. Blood pressure, on the other hand, is the force per unit area exerted on a vessel wall by the contained blood, and it is expressed in millimeters of mercury. So, for example, a blood pressure of 120 mm mercury is equal or equivalent to the pressure exerted by a column of mercury 120 mm high. So, unless stated otherwise, the term blood pressure means systemic arterial blood pressure in the largest arteries near the heart. Pressure gradients. So the pressure gradient is the differences in blood pressure within the vascular system. It provides the driving force that keeps the blood moving, always from an area of higher pressure to an area of lower pressure all throughout the body. Another factor is the resistance. Resistance is defined to as an opposition to flow and is measure of the amount of friction blood encounters as it passes through the vessels. So because most friction is encountered in peripheral or systemic circulation away from the heart, we generally use the term peripheral resistance instead. So there are three important sources of resistance: the blood viscosity, the total blood vessel length, and the blood vessel diameter. Blood viscosity refers to the thickness or stickiness of a fluid. So the greater the viscosity, the greater the resistance. The less easily molecules slide past one another, and it is more difficult to get and keep the fluid moving. So blood is much more viscous than water. Because it contains formed elements and plasma proteins, it flows more slowly under the same conditions. Next is the total blood vessel length. The relationship between total blood vessel length and resistance is straightforward. So the longer the blood vessel, the greater the resistance. For example, an infant's blood vessel lengthens as he or she grows to adulthood. And so, both peripheral resistance and blood pressure also increases. So, blood vessel diameter changes frequently and significantly alters peripheral resistance. So, the answer lies in the principle of fluid flow. So, the fluid close to the wall of a tube or channel is slowed by the friction as it passes along the wall, whereas fluid in the center of the tube flows more freely and faster. So, it means that if there is a decrease in blood vessel diameter, it increases the resistance. In contrast, if there is an increase in blood vessel diameter, there is a decrease in resistance. So now that we have defined this term that summarizes the relationship between them, again, the blood flow is directly proportional to the difference in the blood pressure. So between the 2 points in the circulation. So, thus, if the changes in pressure increases, the blood flow speeds up, and when it decreases, the blood flow declines. So, the blood flow is inversely proportional to the peripheral resistance in the systemic circulation. So if the resistance increases, the blood flow decreases. If the resistance decreases, the blood flow increases. So we can express these relationships by the formula of these two factors influencing the blood flow. The resistance that is far more important than the changes in pressure in influencing local blood flow. So it is because the resistance can easily be changed by altering the blood vessel diameter. So for example, when the arterioles serving a particular tissue dilates, decreasing the resistance, the blood flow to that tissue increases even though the systemic pressure is unchanged or may actually falling. So now let us describe how blood pressure differs from arteries, capillaries, and veins. So we should keep in mind that the blood flows through the blood vessels along a pressure gradient, always moving from a higher pressure area to lower pressure areas. Fundamentally, the pumping action of the heart generates blood flow, and the pressure results when the flow is opposite or opposed by the resistance. As illustrated in this figure, the systemic blood pressure is highest in the aorta, and it declines throughout the pathway to finally reach 0 mm mercury in the right atrium. So the steepest drop in the blood pressure occurs in the arterioles, which offers a great resistance to blood flow. Arterial blood pressure reflects two factors: on how much the elastic arteries close to the heart can stretch, it is their compliance or distensibility, and the volume of the blood forced into them at any time. So if the amount of blood entering and leaving the elastic arteries in a given period were equal, arterial pressure would be constant. But that's not the case. In the body, blood pressure is pulsatile. It rises and it falls in regular fashion in the elastic arteries near the heart. So, as the left ventricle contracts and expels blood into the aorta, it imparts kinetic energy to the blood, which stretches the elastic aorta, and as aortic pressure reaches its peak, the aorta will open. So, this pressure peak generated by the ventricular contraction is called the systolic pressure. Systolic pressure averages 120 mm mercury in healthy adults. The blood moves forward into the arterial bed because the pressure in the aorta is higher than the pressure in more distal vessels. So during diastole, the aortic valve closes, preventing blood flow back into the heart. The walls of the aorta recoil, meaning going back to their original shape and size, maintaining sufficient pressure to keep the blood flowing. So although recoiling, they maintain sufficient pressure. So during this time, aortic pressure drops to its lowest level, which we call as diastolic pressure. And this is approximately 70 to 80 mm mercury in a healthy adult. The difference between systolic pressure and diastolic pressure is what we call pulse pressure. It is felt as a throbbing pulsation in an artery, so as a pulse during systole, and as ventricular contraction forces blood into elastic arteries and expands them, increased stroke volume and faster blood ejection from the heart as a result of increased contractility raises pulse pressure temporarily. So again, pulse pressure is the difference between systolic and diastolic pressure. Now we move on to the capillary blood pressure. So by the time that the blood reaches the capillary, blood pressure has already dropped approximately 35 mm mercury, and by the end of the capillary beds is only about 17 mm mercury. So the low capillary pressure are very desirable. So why? It's because capillaries are very fragile and high pressures would rupture them, and most of capillaries are extremely permeable. Thus, even the low capillary pressure can force solute containing fluids out of the bloodstream into interstitial space. So if there is a presence of high pressure in the capillary, it can move out all the solute going out from the bloodstream. So unlike the arterial pressure, which pulsates with each contraction of left ventricle, venous blood pressure is steady and changes very little during cardiac cycle. Despite the structural modification of the vein, that they have large lumens, there are presence of valves in some of the veins, venous pressure is normally too low to promote adequate venous return. So, if the venous pressure is normally low, how the blood flows back to the heart? So, for this reason, three functional adaptations are critically important for the venous return. First is the muscular pump. So, the muscular pump consists of the skeletal muscle activity. When contracting skeletal muscle press against a vein, they force open the valves proximal to the area of contraction, and blood is propelled towards the heart. So, the back-flowing blood closes the valves distal to the area of contraction. So, people who earn their living in standing professions such as hairdressers and also some of the dentists often have swollen ankles because blood pools in their feet and legs. So instead, standing for prolonged periods may cause fainting because skeletal muscle inactivity reduces venous return. So the next modification is respiratory pump. So the respiratory pump moves blood up towards the heart as pressure changes in the ventral body cavity during breathing. So as we inhale, abdominal pressure increases, squeezing local veins and forcing blood towards the heart. At the same time, the pressure in the chest decreases, allowing thoracic veins to expand and speeding blood entry into the right atrium. Next is the sympathetic venoconstriction. So this sympathetic venoconstriction reduces the volume of the vein, blood in the veins, the capacitance vessel. So as the layer of smooth muscle around the veins constrict under sympathetic control, the venous volume is reduced and blood is pushed towards the heart. So all of these three functional adaptation increases venous returns, which increases stroke volume. That is according to the Frank-Starling mechanism, and therefore increases cardiac output. So maintaining a steady flow of blood from the heart to the toes is very vital for organs to function properly. So central among the homeostatic mechanisms that regulate cardiovascular dynamics are those that maintain blood pressure, principally the cardiac output, peripheral resistance, and blood volume. So there are short-term and long-term regulation. So short-term regulation by the nervous system and also by the hormones in the blood. So first we will focus on the short-term regulation caused by the nervous system. So the neural controls of peripheral resistance are directed at two main goals. First, under conditions of low blood volume, all vessels except those supplying the heart and the brain are constricted to allow much blood as much as possible to flow to those vital organs. Second, altering blood distribution to respond to specific demands of various organs. For example, during exercise, blood is shunted temporarily from digestive organs to skeletal muscle. So to understand fully, let us look at this picture. So how does the body regulates, short-term regulates blood pressure through the nervous system? So if there is a decrease in blood pressure in the body, it will activate cardiac centers located in the medulla. So the cardiac centers in the medulla will increase sympathetic activity and decrease parasympathetic activity. So the increase in sympathetic activity will cause increased epinephrine in the blood. This epinephrine increases contractility of cardiac muscle, thus decreasing end systolic volume. When we decrease end systolic volume, the stroke volume increases. Why? Because the formula in getting the stroke volume is end diastolic volume minus end systolic volume. So, decreased parasympathetic activity on the other hand increases heart rate. Thus, if we increase heart rate, we increase the cardiac output and increase the blood pressure. So, also during exercise, it increases activity of respiratory pump, increases activity of muscular pump, and increase sympathetic venoconstriction, thus increasing venous return that will eventually increase end diastolic volume, and increase in end diastolic volume will cause an increase in stroke volume, thus increasing cardiac output and increasing blood pressure. Most neural controls operate via reflex arcs involving baroreceptors and chemoreceptors. So, when the blood pressure rises, it activates the baroreceptors. These stretch receptors are located in the carotid sinuses, which are dilations in the internal carotid arteries that provides the major blood supply to the brain. Also, they are located in the aortic arch and in the walls of nearly every large arteries of the neck and the thorax. So again, when the blood pressure increases, the baroreceptors are stretched. So this send a rapid stream of impulses to cardiovascular centers and stimulating cardioinhibitory center, thus result into the decrease in blood pressure. So decrease in blood pressure happens because there is a decrease in vasomotor impulses that allows vasodilation, thus decreasing the resistance and decreasing the blood pressure. So when the carbon dioxide level rises, the pH falls, and the oxygen content of the blood drops sharply, the chemoreceptors, on the other hand, located in the aortic arch and large arteries of the neck, transmit impulses to cardioaccelerory center, which then increases cardiac output, and to the vasomotor center, which causes reflex vasoconstriction. So, this increase in cardiac output and increase in vasoconstriction, thus increasing blood pressure. That follows speeds the return of blood to the heart and the lungs. So, the most prominent chemoreceptors are found in the carotid and aortic bodies located close by the baroreceptors in the carotid sinuses and aortic arch. So, chemoreceptors play a much larger role in regulating respiratory rate than blood pressure. The short-term regulation of blood pressure caused by hormones. So, hormones also help regulate blood pressure both in short term via changes in peripheral resistance and in long term via changes in blood volume. So, adrenal medulla hormones such as epinephrine and norepinephrine, during periods of stress, this adrenal gland releases these hormones to the blood. So, both hormones enhance the sympathetic response by increasing cardiac output and increasing peripheral resistance. So, they increase cardiac output by increasing the heart rate and contractility of the heart muscles, and they increase peripheral resistance because they promote vasoconstriction. Next hormone is angiotensin II. So, when blood pressure or blood volumes are low, the kidney releases renin. So, renin acts as an enzyme, ultimately generating angiotensin II, which stimulates increased vasoconstriction. So, it also stimulates release of aldosterone and ADH, which acts as a long-term regulation of blood pressure by enhancing the blood volume. And I will explain that later. So again, angiotensin II increases peripheral resistance by promoting vasoconstriction. Next is the atrial natriuretic peptide or ANP. So the atria of the heart produces this hormone, which leads to the reduction in blood volume and blood pressure. So it decreases blood pressure by decreasing peripheral resistance through vasodilation. So, it generally causes vasodilation. So, the ANP antagonizes aldosterone and products of the kidney to excrete more sodium and water from the body, thus reducing the blood volume. Renal mechanisms mediate this long-term regulation. So as we have noted earlier, blood volume is a major determinant of cardiac output, and it influences venous returns, and diastolic volume, and stroke volume. So if there is an increase in blood volume, it is followed by a rise in blood pressure. And anything that increases blood volume, such as excessive salt intake, which promotes water retention, increases mean arterial pressure because of the greater fluid load in the vascular tree. So, how does the kidneys regulate the blood pressure in long term? So, the kidneys act both directly and indirectly to regulate arterial pressure and to provide major long-term mechanisms of blood pressure control. So, let's focus on the direct renal mechanism. The direct renal mechanisms alter blood volume independently of hormones. So, direct renal mechanism work this way. When there is a decreased blood pressure, there will be a decreased blood flow going to the kidneys. Thus, there will be a decrease in filtration rate going on the kidneys. So, if there is a decrease in filtration rate, it can cause decreased urine formation. And this decreased urine formation increases blood volume, thus increasing blood pressure. Next, we go now to the indirect renal mechanism. So, the kidneys indirectly regulates blood pressure through renin-angiotensin-aldosterone mechanism. So, when the blood pressure declines, the certain cells in the kidney release the enzyme renin into the blood. So, the renin enzymatically cleaves angiotensinogen, a plasma protein made by the liver, converting it to angiotensin 1, and in turn, angiotensin converting enzyme or ACE converts angiotensin 1 to angiotensin 2. So, ACE activity is associated with the capillary endothelium in various body tissues, particularly located in the lungs. So, the angiotensin 2 acts in 4 ways to stabilize the arterial blood pressure and extracellular blood fluid volume. So, first, it stimulates the adrenal cortex to secrete aldosterone. This hormone is the one that enhances renal reabsorption of sodium. So, as the sodium moves into the bloodstream, the water follows, which conserves blood volume. In addition, angiotensin 2 directly stimulates the sodium reabsorption by the kidneys. It prods the posterior pituitary gland to release ADH or antidiuretic hormone, also called as vasopressin, that promotes more water reabsorption by the kidneys. It also triggers the sensation of thirst by activating the hypothalamic thirst centers. So this encourages water consumption, ultimately restoring blood volume and so as the blood pressure. And lastly, the angiotensin 2 promotes vasoconstriction. So it is a potent vasoconstrictor. It increases blood pressure by increasing the peripheral resistance. As I end this video, I would just like to share this quote to you. Knowledge isn't power until it is applied. So I hope that you have learned something today and that you may be able to apply this in your journey to becoming a great dentist. Thank you for listening and God bless.